Welcome, everybody. Thank you for coming to our webinar. As you all know, everyone is welcome to our webinars, but if you please could donate a few bucks to us because we have expenses like Zoom and Meetup and everything. So just go to our website, RockyMountainAtheist.ca, and you can donate any amount, anything, help us keep on going. So just to remind you that we are an inclusive community of atheists, humanists, skeptics, and secular freethinkers, and we welcome anyone to our webinars. We have never had any disruptive attendees, so please let's keep this record clean. Okay, discussion is encouraged as long as everyone shows respect for one another. At the end of the presentation, we will have a Q&A, so if you want to ask a question, just use the hand raising icon at the bottom of the screen where you have the react button, okay? And then we'll call you to ask your question. Quick disclaimer. So the views and opinions expressed by speakers and audience members during this event are their own and they do not reflect the official policies, positions, or views of RMA. So let's see our presentation with our speaker, David Miles. He's from the London School of Hygiene and Tropical Medicine, and he'll be talking about influenza, from the Spanish lady to the winter sniffle. So now let me stop sharing here so David can share his screen with us. Yeah, your screen is okay, David.
Shall I start?
Yes.
Okay. Thank you for coming, and thank you, Helio, for the introduction. I'll briefly introduce myself. My name is David Miles, and my research background is in infectious disease immunology. That is how the immune system protects us against the microbes that cause disease, or pathogens as we call them, and also how it interacts, how it responds to the vaccines we use to give ourselves immunity against those pathogens. And I'm going to talk about one infectious disease in particular today, influenza, which has been with us for at least the last 5 centuries and probably substantially longer than that. It's just that we don't really have good enough records to be sure if we go back earlier than that. So I'm going to start the story around around near enough 5 centuries ago, in the year 1580, when English soldiers were, in the best traditions of the British Army, waging war in Ireland. And we get this account. Suddenly, such a sickness came among the soldiers, which took them in the head that at one instant there were above 300 of them sick. And for 3 days they lay as dead stocks, looking still when they should die. But yet, such was the good will of God that few died. Now, this is probably among the first accounts of an influenza pandemic. We don't know that for a fact. Nobody was isolating viruses in 1580, but there are several aspects of this that really do that are characteristic of pandemic influenza. The first being that this account syncs up with several very similar accounts from other parts of the world at the same time, which implies this was in fact a pandemic, but also the characteristics of the disease. The fact that it seems to have spread among this army so quickly that at one instant there were above 300 of them sick. They then remained very ill for a few days, and then they either died or they recovered equally quickly. And that rapid spread, rapid onset, and then rapid recovery are characteristic of pandemic influenza. In fact, for a long time, they were the defining feature of influenza, and they still remain highly relevant today because influenza doesn't only come in pandemics. Influenza also comes in the form of the seasonal influenza that tends to hit the Northern Hemisphere every winter. And this rapid spread is the reason that it puts so much of a strain on the health service every year, because um, with seasonal influenza, most people don't get that ill from it. Most people will, at worst, get something similar to a cold. But there will be a small percentage of people who become seriously ill and need to go to hospital. And because influenza spreads and affects people so rapidly, everyone who needs to go to hospital needs to go to hospital at the same time. And it's usually at the time when a lot of the hospital staff are off sick because they've got the flu as well. Most of them won't have it that seriously. Most of them will just need to go home, spend a few days to rest and recover. But it does mean that the hospitals are understaffed at precisely the moment when everybody who needs them with influenza is coming in. Now, that rapid onset was the defining feature of influenza for, well, starting in the 16th century, but for several centuries after that. In fact, the word influenza comes from around the same time as that account that I just read out to you. It originated in Italy somewhere at some point in the 16th century. It means influence because these apparently inexplicable epidemics were attributed to being under the influence of the stars. And for several centuries after that, that astrological explanation was really as good an explanation as anybody had. People knew influenza when they saw it, and they were able to distinguish it from other diseases that might have similar symptoms because of that rapid onset, but they didn't really know what caused it until nobody really had much of an inkling until the late 19th century, when we see the rise of germ theory, which was based on the discovery that many human diseases are caused, in fact, by not by alignments of the stars or imbalances of the humors, but by microbes. There was an explosion of discoveries at that time of different microbes, different diseases being attributed to different microbes. Some of them were right, some of them were wrong. And one that unfortunately they got wrong initially was influenza. A German doctor called Richard Pfeiffer, during the 1889 pandemic, dissected the corpses, the cadavers of several people who had died of influenza. He found the same bacterium in all their lungs, and he concluded this bacterium was the cause of influenza. He was wrong, we know now. It was not the main cause of influenza, although for reasons I'll get to, um, he was understandably wrong. But that did define the understanding of influenza moving into the 20th century. It was recognized mostly as being a rapid onset infectious disease that occasionally causes pandemics, actually probably around 2 or 3 pandemics every century, but mostly it simply appears in the winter and causes what we now know as seasonal influenza or winter flu. That was the state of knowledge. Hello? Right. That was the state of knowledge in March 1918, when a young soldier in a US Army training camp called Camp Funston in Kansas reported sick with influenza. The medics initially weren't that concerned about that. When you bring in large numbers of young men from all over the country and put them in an overcrowded training camp, well, diseases like influenza are going to spread. March was perhaps a little bit late in the year for seasonal influenza, but it wasn't that unusual. They weren't so relaxed a few days later because this disease, this influenza, was spreading as fast as it had back in 1580 among the English soldiers. Back then, they said at one instant there were above 300 of them sick. In 1918, it was more like at one instant there were several thousand of them sick. And that photograph there is one of the makeshift infirmaries in Camp Funston. You can see rows and rows of beds as these military recruits were being given really the best influenza treatment at the time, which was to be put to bed and hope for the best. The disease, this influenza didn't stay in Camp Funston. It spread across the United States to the East Coast, and from there, probably by troop ship to Europe and to the rest of the world. This being the middle of the first, this being early in the American involvement in the First World War. It spread very quickly around the world in those few months of, well, the Northern Hemisphere spring of 1918, and it made very large numbers of people ill. It was the beginning of what was to become known as the Spanish influenza pandemic, or the Spanish lady as it was sometimes called. That name wasn't widely used at the time. That name really came up later. Um, Around the time during that first wave, most people simply called it over whichever group of people they didn't like. So in Italy, it was the German disease. In Germany, it was the Russian pest. In China, sorry, in Russia, it was the Chinese disease. In Polish, it was the Bolshevik disease. In Afrikaans, which is one of the languages of white South Africans, it was given a name that we might politely translate as black man's disease. And in most of Africa, it was white man's disease. You get the general idea. That first wave that happened in the Northern Hemisphere spring of 1918 was the first of three waves. Um, the second one being in the Northern Hemisphere autumn, and the third wave happening early in 1919. By far the most lethal of those waves was the second wave that spread across the world in August to December 1918. Through the course of the pandemic, around 1 in every 40 people who caught the 1918 influenza died of it, mostly in that August to December wave. And at this time, it was only people who lived in very isolated communities who didn't catch it. That is 1 in 40 of the large majority of people who were alive in the world at the time. It killed between 50 and 100 million people. We don't know the exact figure because the records at that time are patchy. But to put that into context, the what we're saying here is that the lowest estimate for the number of people who were killed by the 1918 pandemic, which in fact lasted less than a year, is higher than the highest estimate of people who were killed in the 4 years of the First World War. One of those people is worth flagging now, and the direct effect of one of those deaths, in fact, we still live with today. That death was a man called Frederick Trump Sr., who was a fairly successful businessman in New York. Successful enough that he had taken out a large life insurance policy on himself, which led to a substantial cash payment to his widow and to his son, Frederick Trump Jr., who invested that payment in a property portfolio, which it turned out he was quite good at property investment. His portfolio turned into a property empire, which he bequeathed to his son, a fellow you might have heard of called Donald Trump. This is perhaps one of the best arguments I've ever run across for pandemic control or for influenza control. But that aside, after that third wave in early 1919, the influenza pandemic simply appeared to fade away. It didn't come back. It simply went, leaving 50 to 100 million people dead in its wake. And it disappeared not only from communities and hospital wards, but apparently from cultural memory, which is one of the more remarkable factors of the influenza of that particular influenza pandemic. It's been called the Great Forgetting because if you read the history of that time or the literature of that time, you'll find a lot of discussion of the great political events of the world, the First World War, the Russian Revolution, the collapse of the Republic of China, all of which happened at about that time. Where the pandemic where the pandemic is mentioned at all, it's usually only in passing in a brief few paragraphs, which is quite remarkable when you consider the scope of the harm that it did, of the huge numbers of people that it killed. Which raises the question of why and how did this great forgetting happen? Well, one explanation comes from the great American commentator H.L. Mencken, who wrote decades later in 1956, "The human mind always tries to expunge the intolerable from memory, just as it tries to conceal it while current." He was suggesting that the experience of the pandemic was so traumatic that everybody who lived through it simply blotted it out of their minds, even while it was happening. Perhaps, but I'd like to suggest an alternative explanation that's rooted in how history was understood and studied at that time. In the early 20th century, throughout the first half of the 20th century, History was very much the story of great events and the great men, and it was nearly always men who shaped them. It was, if you like, the story of generals fighting their battles, presidents fighting their elections, and chaotic royal successions. It was, or as historian Arnold Toynbee caricatured that approach to history, it was the story of one damned thing after another. That changed in the 1960s with the rise of a new discipline called social history. Social historians were less interested in the great men who shaped the destinies of nations than in the ordinary people who made up the nations whose destiny was being shaped. Not so much the history of generals and presidents and kings as of machinists and farmhands and housewives. And in 1918, we have to include the conscripted soldier. One of the first social historians was an Englishman called Richard Collier. In And in the early part of the early 1970s, he decided to apply a an approach rooted in social history to the history of the Spanish influenza pandemic, or the 1918 influenza pandemic, we should call it. It really didn't have much to do with Spain. He put out a general call for reminiscences of that time. Now, this was more than 50 years after the event, so he probably didn't expect to get much in the way of a reply. But he was inundated with first-hand accounts from people who'd lived through it, or with letters or diaries of people's parents or grandparents who remembered that time and who had spoken of it to their own children or their grandchildren. Because thanks to Collier, we know that the 1918 pandemic was never forgotten at all. And we also know some of the stories of the people who lived through it. It's thanks to Collier curating these accounts into his book, The Plague of the Spanish Lady, that we know the story, for example, of Tarsila Vicenzotto, an Italian woman who roamed the streets of Padua by night during an artillery bombardment, desperately trying to fulfill her husband's last wish to taste an orange for one last time. We know the story of Ilona Molnar, a maid in Budapest whose mistress put her out of the house in case she brought influenza into it. It turned out Ilona was already infected with influenza. As she walked the streets of Budapest, she found herself becoming dizzy and short of breath. She sat down on a bench, and passers-by, recognizing what was happening, called an ambulance. The ambulance took hours to arrive, while a crowd gathered around this obviously ill woman who they were unable to help. And in the middle of a crowd buying snacks from street vendors, Ilona changed color from blue to purple and ultimately died before the ambulance arrived. Another one of those stories comes to us from a reporter called Stock Gison Tonfelt, who was in an infirmary in a barracks stock sorry I beg your pardon was in a makeshift infirmary in an army barracks in Stockholm, when he watched an orderly take a letter to a man lying in a bed. The orderly found the man dead already, but he decided to read the letter to him anyway. It was from the man's mother, urging him to be very careful that he didn't catch influenza and to take good care of yourself and wrap up warm when you go out. It's Collier, of course, only gives us a few isolated tales of people who lived through that time. But it makes me wonder how many of those people were left thinking they were the only ones who remembered the 1918 pandemic, even while they were surrounded by other people who didn't talk about it. It wasn't so much the great forgetting as the great silence. I first read about the Great Forgetting, as it was still called, during the 2020 lockdowns. And I remember thinking, well, of course, that's not going to happen this time. We're all back in 1918, they were distracted by the First World War and the Russian Revolution and everything else that was going on. Of course, we will remember it and we will learn from it properly this time. I was being naive. I should have realized that, of course, the Great Forgetting or the Great Silence or whatever we want to call it applied equally in countries that were neutral throughout the First World War, as in countries that were in the midst of the upheavals of that time. And I do wonder how many people now that we really don't talk about the COVID-19 pandemic very much, are left feeling that we are the only ones who remember, even when we're surrounded by other people feeling the same. Well, there was one group of people who did not forget the 1918 pandemic at the time or later. That group of people was medical researchers, because that pandemic led to a dramatic, because one thing that came out of that pandemic was a change in the understanding of what caused influenza, or at least a challenge to the idea that it was primarily a bacterial disease. That challenge came from two French scientists, both called Charles Nicole and Charles Lebailly, who were actually in Tunisia at the time. They washed out the noses of patients with influenza from the local hospital. They passed those washings through a very fine filter, a filter fine enough to remove any and all bacterial cells. And they squirted the filtered washing up the nose of a few volunteers from the local army base. I suspect the word volunteer may be something of a euphemism in this situation, but those volunteers did go on to develop influenza. Now, at the time, an infectious agent that could pass through a filter fine enough to remove bacterial cells was the working definition of a virus. And it was about as much as was known about viruses at the time, which is due to the different nature of bacteria and viruses, which I'll probably take a moment to explain. A bacterium is fundamentally a small organism. It's made up of it's made up of cells, which, like any other organism, they take in nutrients, they metabolize them, they use it to reproduce, usually by splitting those cells into two. And critically, bacteria were easily visible under a light microscope, even going back to the light microscopes that were and have been known about, in fact, for several centuries, for some time before anyone associated them with disease. A virus is a fundamentally different entity. At its core, it's a piece of rogue genetic code. It has no metabolic processes that it can operate independently. The only way a virus can reproduce is for that code to be inserted into a living cell, from where it can co-opt that cell's metabolic processes to reproduce itself and to make more viruses. When it's outside a cell, it is a completely inert entity. There's an ongoing and frankly rather semantic debate about whether viruses should even be considered alive, which I'm not going to get into. The salient point is that a virus is far too small to be seen under a light microscope, which is why that working definition involved passing it through a very fine filter and see if it could make anyone ill or not. Nicole and Lebailly succeeded in demonstrating that the experiment done by Nicole and Lebailly strongly suggested that they were dealing with a virus, and that the prevailing view that it was in fact that was bacterial was incorrect. Their study was repeated a few weeks later by a Japanese group who somehow persuaded some of their own technicians to volunteer to be infected. These weren't considered to be definitive proof that everyone had been wrong up to that point, though it was pretty good. It kicked off 15 years of controversy, which was ultimately resolved by these two people, two scientists at the British Medical Research Council in London called Wilson Smith and Christopher Andrews. At the time this happened, they were quite a lot younger than they were at the time these pictures were taken. We're talking about 1933, which turned out to be a particularly bad year for seasonal influenza. It felt like you could hardly get on a tram without somebody coughing and sneezing all over you. But Smith and Andrews wondered if this might be an opportunity to finally resolve this debate about what exactly was the pathogen behind influenza. And while they were talking about it, Andrews started to feel a bit peaky. He was sweating, he was shivering, he had a thumping headache. He was coming down with the influenza that the two of them were having a meeting about. Smith, being a good friend and a good colleague, told Andrews he really needed to go home and rest and recover and come back to work when he felt better. But Smith was also a good scientist. So before Andrews went home, Smith persuaded him to gargle some saline solution, which would pick up any microbes in the back of his throat. When Andrews went home, Smith passed that saline solution through one of those very fine filters that I mentioned that removes bacterial cells, and he squirted it up the nose of a few ferrets, which promptly developed influenza. He washed out their noses, he filtered the washings, he put it up the noses of more ferrets. They developed influenza as well. They did a series of experiments, Smith and Andrew, which involved Andrews as well when he was back at work, which proved pretty conclusively that the virus that came out of Andrews's throat was being passed around ferrets and was in fact the root cause of influenza. They clearly established that they were dealing with a viral rather than a bacterial disease. But in 1933, they really couldn't go much further than that. The reason being that, as I said earlier, a virus needs a living cell to replicate itself. And well, the living cells they were using were part of living ferrets, but it's a very cumbersome way to keep a virus alive in the laboratory. And of course, they were getting through a lot of ferrets. Ferrets recover from influenza as fast as humans do, so they needed a new batch of them pretty much every week. But it turned out that what they didn't know at the time was that 1933 was a very good time to make a critical breakthrough in virology or to discover an important new virus. A series of innovations that would revolutionize virology and really were the first steps toward making it the science that we understand today. The first of those steps was a dramatic improvement in how to keep living cells alive, keep cells alive in a laboratory. In fact, the key discovery there was made by Frank McFarlane Burnett in Melbourne, in Australia, who found that there was, in fact, another source of living cells that was a lot easier to keep going, which was a fertilized chicken egg. The embryo inside the chicken egg was, after all, made of living cells. And much to everybody's surprise, and a surprising number of human viruses will infect the cells inside those chick embryo cells. And one of those viruses, it turns out, was influenza. It was the beginning of the science of cell culture, which has led us to the point we've got today, where you can order a whole range of different cells off the shelf and complete with instructions for how to keep them alive, perpetually alive in the laboratory. Another key innovation was the electron microscope, which enabled the visualization of things that are simply much smaller than you can see under a light microscope, including viruses, at least viruses when they're outside the cell, and also the beginnings of the understandings of what genetics meant in the material sense. I said earlier that a virus was a piece of rogue genetic code, is a piece of rogue genetic code. Before there could be any understanding of that, there needed to be an understanding of what genetic code was, which really came in the 1950s as it came to be understood that nucleic acids like DNA and RNA are the molecules that carry the genes of all living things and viruses, depending on whether you include a virus as a living thing or not. All of these discoveries paved the way to being able to ask the question of what is influenza, and to be able to get a reasonable answer back. On one level, we can say that influenza is the pathogen behind it, the influenza virus, which we can see in the left-hand picture there. That is a picture taken with an electron microscope of the influenza virus in the form it takes when it's outside a cell. We cannot see the genetic code in there. What we're seeing is a structure called a capsid, which is made of protein molecules that encase that genetic code and protect it while it's between cells. The capsid really has one function, which is to get that genetic code inside another cell, which it can co-opt to replicate itself as quickly as possible. That's essentially an inert structure. But there's more to influenza than simply the virus behind it. We don't need to be that reductive. Influenza is also the disease it causes, which can manifest in various different ways. But it's also, but that the nature that that disease takes is connected to a whole ecology of influenza, which is touched on in the schematic on the right-hand side there. The key point is that at the center of that diagram is not a human being, it's a bird, because influenza is not primarily a human virus. The major reservoir of the influenza virus is, in fact, the intestines of many different birds. And which raises the question of, well, how can a bird virus, an influenza, or even an intestinal bird virus, become such an effective infection of the human upper respiratory tract, that is the mouth and the nose, um, where we tend to feel that seasonal influenza every winter? Well, the answer is actually with difficulty, because the influenza virus that comes straight out of the back of the bird is not very good at infecting humans. It can infect humans, but it um, the reason it finds it so difficult is that the cells that it can infect are not immediately accessible to it. The cells of the upper respiratory tract of the mouth and the nose are very different to the cells in a bird's intestine. The cells that we have that are most similar to them are in the lung at the bottom of the respiratory tract. And to get to those cells, the influenza virus has to pass its way down the trachea or the windpipe. And part of the function of the trachea between the mouth and the nose and the lungs is to protect the lungs from things going down the windpipe, like nasty bird viruses. It's coated with a layer of mucus, which is full of molecules that are toxic to viruses, and is constantly being wafted up the trachea and into the mouth, from which it gets swallowed down to the stomach, which is, of course, very acidic and will kill any viruses that have made it that far. People do get infected with bird influenza or avian influenza, as they're called, but usually only people who are very highly exposed to it. Typically, people who work on poultry farms where there's an outbreak among the birds. They tend to be exposed to simply so much avian influenza that now and again some virus gets down to the lungs. At that point, there's good news and there's bad news. The good news is that it's as difficult for a virus to get up through the trachea as it was to get down the trachea in the first place. So that person is very unlikely to give anybody else influenza. The bad news is that one place you really do not want an influenza infection is in the lungs. And that has to do with what happens when influenza infects a human. There are two key elements to that. The first is it tends to induce a misdirected immune response that involves a lot of inflammation and doesn't actually do anything to suppress the virus. Now, again, we're all familiar with that. It's the same response we get in the upper respiratory tract whenever we get a cold or when we get seasonal influenza. It leads to a lot of swelling and a lot of very painful and uncomfortable swelling up there. But having that swelling in the upper respiratory tract is not particularly dangerous. It's uncomfortable, it's miserable, it'll put you to bed for a few days, but it's not going to kill you. Having it in the lungs may well kill you because it restricts the ability to accept or to simply to pass oxygen out of the air. It makes it very difficult to breathe. As if that's not bad enough, there's something else that happens when influenza infects, which is it tends to kill the cells that it infects. In fact, and that's bad news when those cells are lining the respiratory tract, because the respiratory tract is not sterile. It is home to a thriving community of bacteria, most of which are harmless commensals that won't do us any harm. Some of them are actively beneficial. But often the difference between a harmless commensal bacteria and a bacterial pathogen that can make us seriously ill is not actually the species of bacterium. It's where in the body it is. A bacterium that is a harmless commensal on the surface of the respiratory tract can become quite dangerous when that surface is stripped away by something like an influenza infection and it's able to infect deeper in the respiratory tract, or even worse, get into the bloodstream. We're all familiar with that to some extent because I think we probably all have experienced having had a viral infection like influenza, which damages the cells in the upper respiratory tract, which is then followed up by a bacterial throat or throat infection or possibly an ear infection. That's how those bacteria get in. That's how a viral infection leads to a bacterial infection. It's bad enough when it's in the throat and the ear. Of course, it's much worse when it's in the lung. It's one of the most common complications of influenza. And coming back to that 1918 pandemic, in fact, these secondary bacterial infections probably caused around, well, were probably the direct cause of more than half the deaths in the 1918 pandemic. This is all very bad news for somebody who gets avian influenza. Um, which of course has been in the news lately because it turns out there's another place where you find cells that avian influenza can infect, which is in the udder of a cow, which is why we're seeing an ongoing outbreak over the last couple of years in US dairy herds recently, which does occasionally affect the people that milk the cattle. Um, Most of those infections appear to be in the eye. They appear to be a very nasty conjunctivitis rather than infections of the lungs, which is moderately good news for because you would much rather because these conjunctivitis can be very nasty, but it's a big improvement than having a viral pneumonia. But it doesn't really answer the question that's probably on your mind at this point, which is how does this virus, how do we end up with these human influenzas that spread so fast that at one instant there were above 300 English soldiers sick, or that spread around the world in a few months, as we saw with the 1918 influenza? To do that, a virus needs to be able to infect the upper respiratory tract, the mouth and the nose, which enables it to get straight into the respiratory droplets that we breathe out all the time and which other people breathe in when we pass them. The route to the human upper respiratory tract goes via the respiratory tract of a pig, because it turns out that the upper respiratory tract of a pig can be infected with both human and avian influenzas. And when two influenza viruses enter a cell at the same time, they go through a process called recombination. Effectively, they form hybrids in which the cells that come out of, because the viruses that come out of that cell will have various mixtures of the genes of the two viruses that went into that cell. They won't necessarily that doesn't make them particularly good viruses. A lot of those hybrid viruses will not be able to infect another cell at all, or they won't replicate very fast. They'll be dead ends. But once in a while, something really nasty comes out of that pig that's been double infected. What comes out of that pig is the influenza virus that causes a pandemic, which is able to infect the upper respiratory tract of a human being, is able to transmit very quickly from one human to another, but is also extremely virulent. It causes a really nasty disease in that human. Part of the reason it can cause that disease is that as well as infecting it's not confined to the upper respiratory tract. It can find its way down to the lung, not by going down the middle of the trachea and running the gauntlet of all that toxic mucus, but simply by infecting cell after cell after cell in the lining of the trachea until it makes its way down to the lung, which means that you've then got an influenza infection causing a large amount of inflammation and stripping cells off the respiratory tract all the way from the nose and the throat down to the lung. You can see why this is a very serious disease. This is what happened in 1918, and it happened again in 1957 and 1968, the other two influenza pandemics of the 20th century, neither of which it turns out were quite as nasty as that 1918 pandemic, but they were bad enough. We saw another new influenza, which did it again in 2009, although really we dodged a bullet there. I think we were extremely fortunate because that virus turned out not to be any particularly more nasty than the seasonal influenzas that we get every winter. We can really thank our lucky stars for that. These viruses spread around the world. They tend to, usually in 2 to 3 waves. And then, as happened in 1919, they sort of drop out of sight and we're left with seasonal influenzas again, which raises the question of, well, where do they go? Well, the answer is they don't go anywhere. They simply lose their virulence. They become the seasonal influenzas that we still get every winter. Why they lose their virulence, they spread just as fast, but why they become less virulent as they do it, we don't know. In fact, there's a lot of things we don't know about influenza. I'm talking mostly about the things we do know today, but let's not lose sight of the fact that that's a long way from being the whole story. Another question you might wonder is why, if we get two or three new influenzas every century and they all become seasonal influenzas, why are we not completely inundated with new seasonal influenzas if this has been going on for centuries? Well, again, the answer to that is another mystery. One thing we do know is whenever a new pandemic influenza appears and mutates into a seasonal influenza, evolves into a seasonal influenza, I should say, one of the existing seasonal influenzas disappears. It becomes extinct. How does one new influenza drive an old one into extinction? Again, we don't know. It'll be very useful to know because it's the only way we know of making an influenza extinct. Well, that is the basis of the virus, the disease, the ecology of influenza. And it leads to the question of, it has led a lot of scientists to the next step, which is trying to do something about it. And most of those things that to be done about it have led to the development of influenza vaccines. In fact, the first influenza vaccines were attempted within a few years of Smith and Andrews first establishing that influenza was a viral disease in the first place. They were pretty simple attempts. Isolate a wild type influenza virus from someone with seasonal influenza, inject it into a chicken egg, culture a load of influenza viruses, kill them and inject them into people. It worked. Those killed viruses contained the protein molecules that make up the capsid we saw earlier, which it turns out are quite important targets for the immune response. Those vaccines induced an immune response, but it didn't really reduce the amount of influenza people were getting because those early influenza vaccinologists promptly ran into a couple of problems. The first was that when the influenza, while recently isolated wild type influenzas will replicate in culture, but they'll replicate slowly in culture. It takes some time to build up enough influenza virus to be useful as a vaccine. And while you're doing that, influenza has not stopped evolving. I said earlier, the pandemic influenzas evolve into seasonal influenzas, but it doesn't stop there. The seasonal influenza is constantly evolving as it passes from one person to another. This causes a problem for the idea of influenza immunity. Now, to start from what happens without a vaccine, it's now spring in the Northern Hemisphere. I'm sure one or two of us here were probably infected with influenza over the winter and will be left with pretty good immunity to the influenza virus that infected you. That virus is now still around in the world, probably in the tropics at this time of year, infecting one person after another and evolving as it goes. It'll return to the Northern Hemisphere in a year or two, but by that time, it will be essentially a different virus to the one that infected you before. Your immunity will no longer protect you against it. Vaccination works by inducing the same long-term immune response or immune memory, if you will, which is induced by infection. And it runs into the same problem. The ongoing evolution of influenza means vaccines get out of date very quickly. And it also means that if you're dealing with a wild type virus that replicates slowly in culture, it's probably evolved. The wild type has probably, by the time you've grown up enough of it to be useful as a vaccine, the wild type's already evolved to the point your vaccine's not going to be much use anymore. This was the problem facing Edwin Kilbourne in the 1960s, who was known for both his virology and his poetry. And I'll read you one of his poems here. He wrote an extract from one of his poems. Human males who get their jollies, spreading trench coats at strange dollies, vanishing without a trace, don't perpetuate the race. It's terrible, isn't it? You can see why we're talking about his virology. But when he applied his virology to influenza, he did it by using the characteristics of influenza against it. One of those characteristics being influenza evolves. So, and not only in the wild, it also evolves in culture. The longer you keep an influenza virus in culture, the more it becomes adapted to culture and the faster it replicates in culture. By the time Kilbourne got interested in this, he had laboratory strains that had been in culture for around 20 years. They replicated extremely fast and extremely rapidly in culture. They were no use as a virus because from the moment they had been taken into culture, their evolution diverged from the wild type. They were now not similar enough to the wild type that was in to any wild type in circulation to induce immunity that would protect anyone against them. But influenza doesn't just evolve. As I said earlier, it also hybridizes. So Kilbourne took wild type influenza that he'd recently isolated and he put it in the same culture as his rapidly replicating laboratory strain. And out of his culture came a set of hybrids of those two viruses from which he was able to select the ones he wanted, which were the ones that replicated very fast, like the laboratory strain, fast enough to be useful for vaccine production, but which expressed the capsid proteins of the recently isolated wild type, and therefore would induce immunity to those capsid proteins. This is the basis for the adult influenza vaccines that we still use today. This is how they're manufactured. Kilbourne simply killed the virus and injected it into people. We now use there's now been another step added to that, which is to purify out those capsid proteins. So it's a much cleaner vaccine. And it works pretty well in adults. It doesn't work so well in children. It's different to the vaccine that's used in children. In fact, that vaccine arose from a different study and from a collaboration across the Iron Curtain in the that happened in the 1980s when the Cold War was still in full swing. This process was started by Galina Alexandrovna of the Leningrad Institute for Experimental Medicine in, well, what's now St. Petersburg. She's something of a woman of mystery. I have not been able to find a picture of her, and I'm not even sure that's her full name. Russian naming conventions being what they are, Alexandrovna sounds more like a patronymic than a surname, but that was the name she used in her papers, so that's the name I'm going to give her. Like Kilbourne, Alexandrovna had found that she could adapt wild-type influenza viruses to replicate rapidly in culture. But she added another step to the process. As well as adapting them to culture, she also adapted them to culture at a low temperature. Human influenza, well, okay, I should start by explaining the human core body temperature, as most of us know, is around 37 degrees. The upper respiratory tract temperature is a bit lower than that, typically around 33 degrees, and that is the temperature at which wild-type influenza replicates best. Alexandrovna placed her cultures at progressively lower temperatures until they were around 28, 29 degrees, substantially lower than the human upper respiratory tract, until she had rapidly replicating influenza that was optimized for much lower temperatures than it was going to encounter in the human body. When she sprayed this virus up the noses of human volunteers, again, not quite sure how she found these volunteers, she found it did not give them influenza. It did not replicate very well at 33 degrees. It was simply too hot for it. It did, however, induce an immune response to the influenza that she'd sprayed up their nose, which is what you want a vaccine to do. In a way, it worked as a vaccine. In a way, it didn't, because, of course, by the time she had gone through the whole process of adapting her isolates to replicate rapidly at low temperature, the wild type had been evolving in a completely different direction, and the immune response induced by her vaccine strain would give no protection at all against influenza infection. The next step of that process was added by Hunain Masab at the University of Michigan, who used Kilbourne's trick of hybridizing a rapidly replicating cold-adapted laboratory strain with a recently isolated wild type, and identifying the hybrid that was safe to spray up somebody's that replicated rapidly in culture, so you could practically make a vaccine out of it, that was adapted to low temperatures, so you could safely spray it up somebody's nose without giving them influenza, but which carried the capsid proteins of the current wild type and therefore induced immunity to it. This vaccine works better in children than the injected vaccine that Kilborn came up with. And in fact, this is the vaccine that children in most high-income countries get every autumn as we go into the influenza season. There's two big advantages to these vaccines. The first is that to both these types of vaccines. The first is they give us immediate protection from the wild type in circulation at the moment. They don't completely protect us from being ill, but they do reduce the severity of that illness. Vaccination could be the difference between the sort of severe influenza that sends you to hospital or perhaps even kills you and getting a nasty cold. Or it can be the difference between getting a really nasty cold and getting a mild cold, or possibly even having an asymptomatic influenza infection that we don't even know we've got. The other advantage of vaccination is that it's possible to combine several different wild type capsid proteins in a single vaccine, which builds up a library of immunity to many different types of influenza and leaves us better prepared for influenza that we may run into often at some time in the future. That's especially true for the childhood vaccine, which is building up a much broader library of immune responses to influenza than they would have if you simply waited for them to get infected every year. That's what vaccination can do. What vaccination can't do is completely protect us against influenza or even completely protect us against severe influenza. And to illustrate that point, I'd like to tell the story of a Yorkshire woman called Lucy. In 2018, Lucy was in her mid-30s with some underlying health conditions, which meant that she got the influenza vaccine, but she was managing them extremely effectively. In fact, she was working as a fitness instructor, which gives you a sense of how fit and healthy she was, how well she was managing her conditions. But 2018 turned out to be a particularly bad year for seasonal influenza. The reason being that as one of the influenza strains had been spreading around the world, it underwent a mutation that led to extremely rapid evolution. So rapid that when it came back to the Northern Hemisphere, hardly anybody had any immunity to it. People who'd been infected before had immunity to whether that immunity came from having been infected by an ancestor of that virus or a vaccine based on that virus, that immunity was to a virus that was very different to the virus that came back and infected everybody, including Lucy. And it hit Lucy very hard. It got into her parasympathetic nervous system, or it affected her parasympathetic nervous system. That is the system by which the brain controls the heart. 5 times that year, she was hospitalized with an irregular heartbeat, or tachycardia, as it's called. She was left with a condition of permanent dysautonomia, which means effectively her brain does not regulate her heartbeat very well. She can't stand up for any length of time without risking passing out. This condition got worse a couple of years ago when she developed COVID-19. She's now reached the stage in her early 40s where she sometimes needs a mobility scooter to leave the house. And Lucy is the reason, or people like Lucy, is the reason why I think it's worth continuing to talk about influenza. Because while I have told the story of discovery of the influenza virus and the influenza disease today, and I've told the story of the successes behind the influenza vaccine, Lucy's story tells us the limits of those successes, and it's a reminder that every year when season comes around, there will be a small number of people who are hit as badly as Lucy was, whose lives are permanently disrupted by that influenza infection. And they are the people, and they are the reason why we need to continue doing influenza research and hopefully to come up with a better solution to influenza than any we have today. That's the thought I'd like to leave you with. So I will say thank you very much, and I would be happy to take any questions.
Okay, thank you very much, David. Let me just. So, Brent, Did you raise your hand?
No, that was a clap.
Oh,
Oh, thank you.
Yeah, excellent though.
Well,
But actually, while I do have the platform, then just FYI, David, our group has a book club, and last month we did The Code Breaker by uh, with focusing on Jennifer Doudna for gene editing. So does that play into the role of immunology with respect to the influenza virus?
Yeah, well, Jennifer Doudna, of course, invented CRISPR-Cas9, which is a method of directly editing the genome. I think we're still at the stage where we're really finding out what the applications of that are, and we're seeing it coming up, being tried out in all sorts of different things. I mean, there is an obvious application in vaccine production going on there, because if you can directly edit the virus's genome, then you can effectively turn a dangerous virus into a harmless virus, or put whatever proteins you want into a carrier virus. At the moment, there is no vaccine that's been made like that that's been licensed. I'm hopeful that we will see some. Obviously, the ideal application of CRISPR-Cas9 would be to directly edit the genome of infected cells, for example, or of damaged cells. But that's a long way in the future at the moment.
Okay, thank you.
okay, Jason, you can go first. Sorry, Jim, you can go first.
there. There. Now I'm not a mutant. So basically, okay, you get people against vaccines making claims about vaccines being dangerous in some way. Now, I suppose nothing is totally 100% safe, but I guess the matter is, how do you make the risks of getting the vaccine much smaller than the risks of not getting the vaccine?
Um, well, yeah, that's something that, of course, vaccine science has been wrestling with for a good couple of 100 years at this point.
Yeah,
I think
yeah.
the answer is that we do have a reasonably established pathway for initially testing a vaccine in animals,
Hmm.
then testing it in progressively large numbers of people with um, very tight surveillance to make to very sorry. I'll start that sentence again. With very close monitoring to assess the harms of that vaccine. And yes, vaccines have been found to be potentially harmful at that stage, and that has led to the discontinuation of the development. And then perhaps the most important stage is that when the vaccine goes into the wider population, it then continues to be monitored for safety, because trials, of course, tend to be thousands or tens of thousands of people who are often selected to exclude people with all sorts of underlying health conditions. As soon as it goes out into the general population, it's now going into millions or tens of millions of people. So you have the problem that you might have very rare adverse events that you simply hadn't looked at enough people in the trials to find, or you may find it interacts in some way with other underlying conditions. And the only way to be confident of that is to have this ongoing process of safety monitoring after the vaccine's gone out into after the vaccine's been licensed, which has worked quite well. We've seen there was a rotavirus vaccine that was used in the United States in 1996, which was established to have certain complications, which led to it being withdrawn. Um, of course, the Oxford AstraZeneca COVID-19 vaccine went through a similar process. It was I think it was initially found in Norway and Denmark that it caused significant side effects, and what could cause a life-threatening blood clotting disorder in some people. It is this ongoing monitoring that makes vaccines safe. Unfortunately, it's this ongoing monitoring that, when it successfully finds a problem, often fuels the idea that all vaccines are dangerous, which can be a very difficult one to address. And I'm bound to say, at the risk of getting slightly political, that if you really want to monitor potential harms from vaccines, absolutely the last thing you do is gut your national public health agency, especially when it's the CDC and is one of the most successful public health agency in the world at assessing vaccine safety. But that appears to be the current US government's approach.
Yes.
Right.
Yes. It seems longer, but it's been only that JFK Jr. has been the head of that department, but it's actually only been a few measly months.
Measly being the appropriate word, yeah.
Mm-hmm. Yeah.
Okay, let me ask you one question here, David. How far are we from a universal influenza vaccine if there's this possibility in the future of not having to change every year?
Yeah, this seems to be one of those things that's always 5 or 10 years away until it actually arrives. There's been quite a lot of research into trying to find some element of the virus that induces an immune response and is stable, while the other parts of, I mean, at the moment we're targeting the immune response on the capsid proteins, which are the bits that probably change most rapidly. It would be great if we could find a target for the immune response that protects us against influenza and does not evolve along with the capsid proteins. So far, nobody's found one, which is not that surprising. I mean, typically, the components of a virus that are most likely to evolve are those that are under evolutionary pressure, and the ones that are under evolutionary pressure are getting that pressure because they're targeted by the immune response. So this is not particularly unlikely. The other approach is simply to layer a vaccine with as many different types of influenza as possible, which is actually feasible, which has been done. The purpose of that would not really be to protect us, to prevent us from ever getting seasonal influenza again. It would be to give us a baseline of immunity so we can handle a future pandemic influenza a lot better. It probably wouldn't stop us getting ill from that pandemic influenza or from getting seasonal influenzas, but it would make us far less likely to be killed by a future pandemic. And one of the advantages of doing it that way is, well, because pandemics appear and spread so fast, it's very unlikely that anybody will be able to come up with a vaccine against that pandemic influenza and distribute it around the world fast enough to prevent at least the first wave or two of that influenza. So an influenza vaccine that could give a baseline of protection against that in advance would have a substantial advantage if you can persuade a lot of people to take it against something that may or may not happen in the future, well, that will happen in the future, but may or may not happen in the next decade or two. When we're talking about practical universal influenza vaccines, that is often what we're talking about, less than something that would replace the annual vaccination program, though.
Okay. And how about the vaccine that doesn't use eggs?
Yeah, we have that. There are influenza vaccines that are cultured actually through genetically modified insect viruses, specifically for people with egg allergies. One possible interim approach would be to move away from killborn. I mean, because influenza, as I said, most influenza vaccines are still made actually in chick embryo cells, not actually in eggs anymore, as the cells in culture. There's pretty good evidence that actually they are pretty safe from people who have egg allergies because there's no egg albumin in there, which is what triggers the allergy, although it's still contraindicated for people with egg allergies. There is some suggestion of moving over to possibly an mRNA vaccine, which would have no element of an egg in it at all, and would actually be a lot quicker and cheaper to produce. It's a nice idea, but of course it would mean that somebody has to test that, or come up with the money to test that on a large scale, which may be the stumbling block. But I mean, we do have a situation at the moment where we're largely dependent on, Kilbourne's approach was 1960s biotechnology, which is pretty cumbersome by modern standards, but we're still using it because that's the approach that's been tried and tested and has gone through vaccine trials. There are arguments for coming up with more modern, more streamlined approaches, but they would have to go through vaccine trials first.
Okay. Any other questions? No? Okay, so just let me share
I
my
believe
screen.
Brent's got his hand up.
Oh, sorry. I covered his. Yeah, go ahead, Brent.
Yeah, a few miscellaneous questions here. Do I understand that the hybridization of the influenza virus has been promoted or has been accentuated by the number of pigs that have been cultured or have been used in food production?
That's an interesting question because logically you would expect that to happen. I mean, it makes sense that it's going to be domestic pigs that are in close proximity to birds and humans at the same time and are going to be the mixing vessel, as it's called, of new influenza viruses. And it makes sense that as there have been more and more people in the world, there are more people keeping pigs, and therefore there are more domestic pigs and more opportunity for this to happen. But we do pretty consistently see 2 to 3 influenza pandemics every century from the 16th at least up to the 20th century. We're not seeing a marked acceleration in that process so far. Now, of course, when we're only talking about 2 or 3 events every century, it may take some time to actually see a marked increase in that, a marked acceleration in the appearance of pandemic influenzas. But yes, I mean, logically, you would expect that we are more at risk of an influenza pandemic now than we ever have been before, purely because of the number of domestic pigs. There is no direct evidence of that at the moment, though I think we shouldn't be ignoring the a priori evidence that I think you're describing there. Did you have another question?
Yeah, it's more a personal question. I'm actually looking at traveling to South America in a few months. So I did take or get the flu virus in the fall, but my understanding is I'd probably want for full coverage. There's probably a high or a newer one in South America now. Is that true for people who travel then if you're going like between North and South America?
Well, I mean, influenza is, of course, constantly evolving. In fact, the way that global surveillance works is that there is a system run by the World Health Organization, which works with, I think, about 120 countries around the world to collect samples of influenza virus and to monitor that process. In the mid to the higher latitudes, we get distinct seasonality. So what will happen is the influenza virus that's causing, well, in a few months, it'll be winter in the southern hemisphere, and it'll be their influenza season. And the virus that's collected from their influenza season will then be used as the basis of the vaccine for the autumn in the northern hemisphere. And then when we do get the influenza arrives in the northern hemisphere, that virus is the basis for the vaccines given in the southern hemisphere, and so on and so forth. What happens in the low latitudes is very confusing. These are countries that don't have the same distinct seasons that we see in the mid to high latitudes, but of course you do usually have some sort of seasonality, and you do have some sort of seasonal pattern in influenza infection, but there's very little consistency of that pattern between one part of the tropics and another. And actually it's not nearly as well studied. So yes, you are likely to meet all the influenza in their sort of intermediate state between the northern and southern hemisphere winters if you go into the tropics. What that means in terms of vaccination is a good question. I think the best thing that you can do at any time is simply have the most up-to-date vaccines available to you. Yeah, I mean, most of the evolution is probably happening in the tropics, purely because we don't see influenza in the mid to high latitudes for very much of the year, but they seem to be in some part of the tropics most of the time. It's not very well monitored and it's not very well understood, which makes it difficult to say what to do. So I think the best you can do is just have the most up-to-date, or always have the most up-to-date influenza vaccine is the best bet.
Okay, thank you. One little curiosity, more so than related, I think, to your topic is you mentioned the social history and Richard Collier. Is that the person who also had the Collier encyclopedias? Like, is that just coincidental?
Ooh, that's a good question. I don't think so. I think the
Okay.
Collier encyclopedias were earlier, but I would not like to. Don't quote me on that. I would need to look that up. He did write quite a few books on social history.
Which is actually, I wasn't aware of that. So very interesting. And thank you for that insight.
Okay,
And
Jim.
that's all for me. Thanks.
Okay, does getting a sort of vaccine against one flu strain mean that your infection from another flu strain is going to be sort of less nasty? Like, does it provide you with some protection?
Well, I mean, first of all, you're never. Sorry, you don't know you hadn't finished.
Okay.
Yeah.
Well, okay. Just does uh, okay. The okay, does the vaccination against one strain provide poor but still some protection against another strain?
Yeah, that's a good question. Probably, though, in practice, you're never going to get a vaccination against only one strain. The vaccine you get it contains all the currently circulating influenza A and influenza B strains that have been identified, which is part of the reason I said it does actually give you much broader base of protection than being infected. Um, is there cross protection between them? That's a really good question. Probably. Some of them are not that different to each other. Um, and of course, we do have this unexplained phenomenon where the appearance of a new influenza strain in the wild will lead to the extinction of another one, which implies that it's somehow inducing immunity that kills off the other one, even if we don't quite understand how. So I'd say it's quite likely it'll give you some cross protection. I'd say that there probably is some sort of cross protection going on, although it's unclear exactly how much. And as I say, if you get the vaccine, then you're going to get all the currently circulating strains anyway.
I recall the claim that some flu strains died out during COVID, and this was attributed to the things we were doing to prevent COVID spread, also preventing the spread of these flu strains.
Right. I'm wincing because I cannot remember. It was the Yamagata lineage of one of and I cannot remember exactly which of the influenza strains it was a lineage of. Um, but yes, this happened because a few years ago, one of the wild types actually split into two different lineages, one of which became known as the Yamagata lineage, which did become extinct during COVID, and yes, was probably made extinct by the lockdown measures that were put in place at that point. Which does raise the question of why that one and only that one, which we don't know the answer to. I mean, it is actually distinctly possible that if it does suggest that if we could, for example, persuade the entire world to go into lockdown for a few weeks at some point, it might well kill off all circulating influenza strains. But obviously, that's not going to happen.
And we'd still get a new strain coming from the birds and the pigs.
Sooner or later, yes, yes, but we might have a few years with that before that happens. But yes, that did happen. Yes.
Yeah. I've read stuff about well, kind of speculative about some of these pandemics that occurred in ancient history, like late second century AD in the Roman Empire, and which disease was it? Was it smallpox? Was it measles? And
yeah, this comes up a lot. I mean, I have to say, I'm not a big fan of this sort of this because it ends up getting very, very. You end up with evidence being very drawn from very tenuous findings, and um, partly because with the descriptions of illnesses being what they are, it's often rather difficult to work out exactly what that person meant, and how to calibrate it against how we would describe diseases now. So yeah, there is this whole hobby that I think some epidemiologists have of looking at accounts of ancient plagues and trying to say what was causing them. But I think it often is rather tenuous what they come up with.
Yeah, one of the things I saw in was that sometimes the symptoms of a given disease sort of shift over time. Is that right?
It can happen, yes. Well, I mean, we see it, of course, we see it with pandemic influenza, which shift from pandemic to season, which shift from pandemic influenza, which can be a much more systemic disease that causes pneumonias, into seasonal influenza, which is usually a cold, which is a classic case of that.
Yeah.
And
So yeah. So the first time smallpox hit Rome or whatever, the symptoms might have been somewhat different from what it became later, and it'd be hard to tell what it was.
yeah, I mean, there were two. There was I think the Antonine plague and the Cyprian plague happened a couple of centuries apart, and I've heard it suggested that one of those was probably the first smallpox, the first time people got smallpox, and the other one was the first time somebody got measles, but nobody seems to know which is which. Well, a lot of people claim to be able to say which is which, but nobody can agree which was which I think does give you a sense of how vague these things are. And I mean, it's worth bearing in mind that there probably were a huge number of diseases that have happened over the past centuries or millennia that simply went extinct for one reason or another, and we don't have them at all now.
Yeah.
So they may have been caused by something that we just don't have anything to compare them to.
Yeah, let's see. Yeah, I recall reading about the sweating sickness or something in 16th century Britain.
Yeah.
That it was there for a while, and nobody knows what it was.
Yeah, and you get accounts of respiratory diseases that seem to be spreading through people and horses at the same time, which could actually be an influenza. It could well be that maybe horses act as a mixing vessel as well as pigs. Who knows? Or it may have been something completely different that we don't really have anymore because we have far fewer people in close contact with horses than we used to. It's just impossible to tell for sure, of course.
Still a lot of mysteries, eh?
I mean, historical epidemiology is quite a discipline if anyone wants to jump into it and is willing to accept that they are never going to come up with any firm conclusions on anything.
Yeah. Yeah. Which, yeah. I recently read a time travel novel where some 21st century people are stranded in the late second century AD Rome, and which disease that plague was is a plot point.
Right. Yes.
Because you can do something about smallpox.
Yeah.
Yeah. Yeah.
Well, you could do something about measles as well if you can do something about smallpox, which it may well have been.
Yeah.
There is reasonable. I have seen some evidence based on genetics that actually measles probably diverged from a livestock disease called rinderpest early in the first millennium CE. So it could have been that which does sync up with the idea that I think it was the Antonine plague could have been measles, or could have been the first time people got measles, but maybe it wasn't. It's equally likely.
Yeah. Anyway.
Yeah.
Okay, any more questions?
Okay.
No? Okay, so let me...
Anybody? More comments?
You're silent there, Helio.
Let me just show the last few slides. Here's the contact about for our speaker, and I'll put them on the video when I publish it, sorry, next week. And now for our next webinar, we'll be having on May 28th at 7 p.m. Information literacy for college students and everyone else with Fiona Dyer.
Yeah.
This one will be very good.
Sure. Okay. Yes. Now I noticed the variolator. Now, wasn't variolations the giving people a a hopefully weak version of smallpox before vaccination was developed.
Yeah. Yes.
Yeah,
Deliberately infecting people with smallpox. Yes.
yeah, and I guess it was less bad than getting disease naturally often enough that it was a persisted.
Well, I mean, the idea was that you took smallpox tends to lead to a lot of skin pustules,
Yeah,
and that you took a pustule and you ground it up and then you inserted it in a cut in somebody's arm, usually,
yeah,
which caused one pustule and left them with lifelong immunity, which did usually work. I mean, they seem to have described it in terms of giving them a weak form of disease. In fact, that's probably not what was happening. What's probably happening was that smallpox tend to initially infect through the upper respiratory tract in the same way as, um, well, the same way as human influenza does, and then spread through the body after that. I mean, what was probably happening was just putting it under somebody's skin, was putting it in a place where it couldn't spread through the body for one reason or another. Um, a, well, it killed fewer people than natural smallpox did anyway. What's that?
uh, this is a book from a few decades ago, Plagues and Peoples,
Peoples.
that
Mm.
I read and got sort of the history of the influence of diseases on human history. Uh, Apparently, it was one of the very earliest books on the topic. Uh,
I've heard of it. I have not read it. But,
yeah, yeah, yeah. Anyway, so that's a fair bit of where I've gotten some information on that, and of course, there's probably a fair bit that has been found to be inaccurate in the succeeding decades. Yeah,
yeah, it's generally, I have to admit, I haven't read it. It is generally credited with the idea, with introducing the idea, or at least one of the early things to introduce the idea that infectious disease has been a major force on human history, which
mhm.
um,
Yeah,
is, I think, possibly a bit more mainstream now,
yeah,
though it's still debated exactly how significant it's been.
yeah, yeah. Well, gee, like if that plague of the Antonine plague killed more Romans than than Germans, it might have weakened them, weakened the
Well,
empire. Yeah,
there is an argument that I think it was the Cyprian plague of about that time, actually led to a sort of cultural devastation of Rome that led to them believing that maybe they were worshipping the wrong gods and turning to Christianity. Um, which, if that's true, that was a pretty significant moment in human history.
yeah,
Uh,
yeah,
I forget if that was a Cyprian or the Antonine plague that happened around that time, but it did seem to happen immediately after one of the major plagues.
yeah. Right. Yeah. Well, my impression is that it's sort of the third century when Christianity sort of really started to take off. And
Yeah, because it was adopted by the Romans,
yeah, right.
um,
Yes. Yeah.
and
Well,
and then distributed
okay.
through the Roman Empire. But yeah,
Yeah. Yeah.
and I cannot remember which of the two plagues it was that happened immediately before that. So,
Yeah. Yeah. Well, there was this 50-year period when there's almost continuous civil war.
Guys, sorry, Jim, to
Yeah.
interrupt. I'm
Yeah.
gonna have to leave here.
okay.
Yeah, almost continuous civil war over
Yeah.
who the emperor, and
Yeah.
that was happened in the third century. Yeah,
Yeah. Yeah.
but how much that had to do with the diseases is another matter.
Well, yeah. I mean, it is, of course, with these things. I mean, I suppose that is the nature of history, is that you get a set of events that are all connected to each other, and every single event will have a whole set of other events that were the cause of it and will influence multiple events in the future. And trying to identify what are the most important through lines is, well, the role of the historians, really, not the infectious disease researchers like me. But
Yeah.
yeah.
Anyway.
Yeah, that's
Yeah.
fascinating. This topic is fantastic. David, thank you very much. It was great having you here. I'm glad that we managed to find some time that could fit both of our schedules. And next week I'll be broadcasting this to everybody.
Thank you.
So I'm sure that people will enjoy. And we had people asking on Meetup when they can watch, because this is not a very common arrangement that we have, where we have the recording and broadcasting. It started just this year. So, but it seems to be successful, and it's very, it's important to have this kind of information in a way that we can spread easily. Thank you very much. It was fantastic.
And you've got my social media links there. Just if anyone has any questions, I'd be happy to take them as long as people know where to find me.
Perfect, great. Yeah,
Or through my website is fine as well.
Yeah, I'll post when I post the video. The video will be private until next week. And then next week I will put it on our social media, and then I'll send you the links again the same way I did
Thank you.
the post here. So you'll have a way to share with your social media as well.
Thank you. Thank you. I'd appreciate that.
Thank you, everybody. It was fantastic. See you next time.
Okay. Thank you. Bye.
Bye. Bye.